Hi, everyone. I'd like to welcome you all to the HUPO Education and Training Committee webinar series. The current series that we're doing right now is on proteomics technologies. So today, Philip Remeš is going to be our speaker. Philip is a research scientist at Thermo Fisher Scientific. He has his Ph.D. from UNC Chapel Hill with Gary Glish, and today he's going to talk about the past, present, and future of ion trap-based mass spectrometry. Just so that everyone knows, these videos get recorded and they get posted to YouTube afterwards, so we can share them with other people. Everything is recorded freely shared. I will write down questions from the chat, and I will ask them to Philip as they come up. And so with that, Philip, take it away.
All right. Thank you very much, Brian. I am very happy to be speaking to you guys today about this topic, which is near and dear to my heart. And I started my grad school in 2003, kind of when ion traps and proteomics were taking off, as we'll see. And ion traps have really been there since mass spectrometry proteomics started to get big. And we're going to have a brief overview of how ion traps work. We're going to see how the strengths of the traps, they work really well in proteomics, and the weaknesses are often not as important as you might think they are, especially for bottom-up proteomics. The way that they've been used is changing from going to almost purely qualitative to more quantitative, and we'll end up looking at the state of the art right now and where we might go in the future, and whether ion traps could still be there. So quadruple ion traps and quadruple mass filters were invented in the 50s, and the scientist that gets the most credit is usually Wolfgang Paul, and he shared the Nobel Prize with others for his work in 1989. And so these devices, they were very commonly used in the physics community to trap ions and manipulate them. And curiously, for those looking back today, they were used in kind of an inefficient way, and very analogous to a quadruple mass filter, where ions were put into the trap. The one that you wanted to look at, you would isolate that one, get rid of everybody else, then you dump it out to a detector. So it was kind of a serial process. We can have a little look at how the ion traps work, how the quadruple mass filters work, and kind of get the basics. We know that like in a quadruple mass filter or linear ion trap, you could have 4 rods, and you would apply a high voltage in the thousands of volts. That would be in the radio frequency range around a megahertz. We often call this an RF voltage. And it's a very dynamic thing. So at one snapshot, the ions would be being pushed, squeezed in one dimension, and they'd be falling off this hill and the other towards the electrodes. And at another snapshot, the opposite thing would be happening. And if you rotate this field at the right frequency, you can trap ions and get them so that they kind of go around. They never hit the electrodes. And the ions, they're oscillating back and forth with these characteristic frequencies that depend on their mass to charge. And interesting property of the traps is that their frequencies, they change as you change the amplitude of that trapping voltage, of that RF voltage. So at some low potential, the ions would have some low frequency, and at some higher potential, the main frequency would be higher. So this property, it was exploited by George Stafford he came up with it at ASMS in the 70s, but in the 80s is when it really got developed with the help of lots of luminaries here at Finnegan at the time, like John Syka. And here's a figure from their patent where they were ramping the main RF voltage, causing all the ion frequencies to shift, and then they would one by one fall out of the trap and be detected. And this was much more efficient, as we'll see, and it led to the first commercial ion trap. This is a video from a simulation, in-house simulation program that we have. It has some graphic card acceleration to go very fast, and we're looking at mass analysis of 2 different ions, one ion and an ion that's just a little bit larger than it. And you saw there at the end, the blue ones came out first, then the red ones came out later. And that was just a snapshot of a few tens of microseconds. And often people will show these stability diagrams. What was happening is as we were ramping them over here towards this the supplementary excitation that's on these X electrodes. So as I say, the ion traps, they were there at the beginning of when proteomics got big, or mass spectrometry got big in proteomics. And if you look at how the publications were starting to increase in that field with these search terms, they were both just kicking off right then. And Aebersold and Mann in their review in 2003, they said that because ion traps were so robust and sensitive and inexpensive, they produced much of the data in the literature. And what were people trying to do back then? Because it's changed a little bit. As they said, the outcome of the experiments was trying to figure out what's in the sample, what's the identity of the peptides. So these were mostly qualitative experiments, and they were smaller scale. So a typical experiment would be to do some fractionation step, then do some gel electrophoresis step, and then cut out regions that you might be interested in, do a digestion, then do another separation with the LC, do ionization, and do the data-dependent acquisition experiment where you do a survey, you pick out intense precursors, and then you try to sequence them. And this sequencing still sometimes was done manually in some hardcore labs, or sequest had been invented already a decade earlier. And people were still arguing about the best way to score these spectra to figure out if they were good matches or not. So these were qualitative times, and the principal, I could say maybe competitor, I mean, there were TOFs, maybe not quite as reliable at the time, but the principal competitor was the triple quad. And for a qualitative experiment, it's just not as good, because if you're going to look at a particular master charge, you have to scan over a range. As you look at one master charge, you're not looking at every other one. So for a 1,000 Thomson scan range, your duty cycle is 0.1%. And yet still, Eng and Yates in their Sequest paper in 94, they were using a triple quad with a 2-second scan, but this just wasn't very sensitive. Ion traps, on the other hand, now, if we look, this is a little bit more complicated schematic. Sometimes we would call this like a scan function. It's doing a few different things, and it's all doing them in the same space. You're injecting ions into it, you're isolating the precursor you want, then you're fragmenting it, and then you're analyzing it. And a typical experiment at the time would take about 200 milliseconds. And this process was about 2.5%, which isn't very high, but was like 25 times more sensitive than the triple quad at the time. So the big problem was that these ion traps, these 3D types, they have this huge voltage, thousands of volts on this ring electrode. And so it's not very easy to get the ions come into there and to be trapped. It can only be done for a very small fraction of the trapping cycle, maybe 5% or so. So this is where you get from something that could be 50% efficient, you get it down to 5% or so. But still, it was really good for what they were doing, and you see the rise in the publications using different kinds of 3D ion traps, in particular ours in the mid 2000s, starts to rise. And so one of my colleagues said, you need more jokes. You need to kind of break it up so people don't fall asleep. So I hope that you guys are doing okay. We're going to keep on talking about the ion trap sort of qualities now, or their perceived weaknesses, actually. So what people said about them then, and I'll have my rebuttals. And some of these things are the same things that they'll say today, and you might have questions about. So maybe I'll preempt you. So the one thing that they said was that if you just have a standalone ion trap, the type of activation that was done was not the same as a triple quad, which is done in a collision cell. Or you could call it like a beam type of activation. Here in the ion trap, it was a resonance type. So you're just resonating the precursor that you want with this small little voltage that's applied to the X electrodes. And because of the way these traps worked, the activation was done at a certain low frequency, so that you could still excite them, but you could keep them in the trap. But this imposed this so-called low mass cutoff thing that would happen at about a third of the precursor. So based on this parameter for the precursor being 0.25 and seeing that every mass that's formed less than or that's greater than 0.908 is not trapped, there's like about a third of a cutoff, where like a collision cell activation has a cutoff that's more in the range of 0.1 or so. So it's like 2 to 3 times better in that regards. However, for peptides, it's like it's perfectly made for the ion trap. It didn't really matter because you're mostly looking at 2 to 3 plus ions, and these then they fragment into mostly 1 plus ions, which are often bigger than the precursor. The precursor here is like 750. So most of these are bigger. And really in proteomics, the smallest informative fragment is a Y2, because if it's tryptic, you have the lysine or the arginine. It doesn't tell you a whole lot. So in most of the range, if you look at what the low mass cutoff is, you could see down to the Y2 all the way up to like 750 or so. So from that perspective, it didn't matter for proteomics, and resonance CID was fine. You could make an argument that for small molecules, it might not be as good some of the time. Especially you could have some more interferences. Like when we were looking at ASMS some time ago, we were looking at resonance CID for pesticides. And here, if you made a histogram of where did the fragments fall relative to the precursor, you're getting lots of water losses, lots of like common moieties falling off. And since it's a resonance process, they're not excited anymore like they are in a collision cell and to further fragments. And so the beam type CID would give you more of a unique signature, like you can see for this. Here I have the colors reversed accidentally, but the beam type CID is a little bit more unique. Of course, you could just do what SciEx did originally with their Q-trap and what we did later with the Tribrid and with our new mass spectrometer, the Stellar. You could have a quadruple mass filter to do the precursor selection. Then you could have a collision cell, and then you could have your linear ion trap. So the next, and maybe the biggest thing that people might say is, how could you do proteomics with this low resolution? And Aebersold and Mann, they pointed out that this is one of the disadvantages relative to, at the time, the TOFs or the sectors or the FD instruments. And Cook said, yeah, this is what we're looking forward to, that we should make these exact massive measurements. And I can say, like, as an ion trap scientist coming into the field, this is kind of your dream. You want to, we all wanted to make their peaks skinnier and make them more accurate. And unfortunately, we haven't really succeeded. Our specs are about the same as they were back then, like plus or minus 0.1 when you've calibrated. And because of reasons you're going to see, like there's still some mass drift. Like if in an experiment, you probably use a larger tolerance than this on your search. But this is actually okay. And the resolution that we could get back then is maybe about the same, on the order of 10,000 or so. And an interesting fact about that is that the ion traps are already punching above their weights in terms of the resolution. Alexander Makarov in the mid 90s, he noticed this curious thing, that the resolution you could get when you scan slow, it was like 10 times better than the theory was predicting. And he showed that this was because our traps in particular had a particular geometries that we called them stretched. And this introduced steep fields, higher order fields, they could be called, that were higher than quadrupolar. And this caused the resonance to be sort of asymmetric, so that when an ion came, approached this resonance from the right direction, it would get excited very fast and get ejected really fast. So you could get higher resolution than expected. And if you look at the resolution that you get per time, looking at like how long does an ion spend being in resonance and what's the resolution that you get, like with the very slowest scan rates, you can get above 10,000 easily. And that's like faster per time than you get in an Orbitrap, of course. An Orbitrap gives you this resolution for all of the ions at once. You have the FD advantage, whereas the linear ion traps really, I would say, too slow to effectively use the slowest scan rates. Well, let's look in proteomics, how much resolution do you need? How much mass accuracy? We did an experiment like 10 years ago where we had one of our hybrid instruments, our Tribrids actually, and we had the exact mass measurements for the Orbitrap, and we had the ion trap MS2 spectra, and we said, what happens to our search results, the number of peptides we can confidently identify as we increase the tolerance from 10 ppm, kind of the standard for the precursor. It's like 0.005 at master charge 500. You can see the IDs, they're falling off down to eventually about 13%, but then they plateau. So that's pretty interesting. Why don't they just keep on going down? You can still do a pretty good job, missing maybe 80, missing 13% of them with a nominal mass precursor measurement. And Senko and Scott Gerber, when they were playing with, I think this was from the LTQ FT, they noticed a similar thing with even accurate mass measurements, where if they would kind of degrade them by truncating the size of the FTs, they could get good search results and not get a loss in the IDs down to only like 512 points in the whole spectrum. So the resolution was pretty low. I think the reason that this happens is that peptide masses, they're not distributed evenly, like in a uniform distribution with mass to charge. They're in these isolated little islands. And this was pointed out by several people. There was an interesting review by Muddyman, and I think it was Mann that first pointed it out at ASMS. And this is because of the mass defects of carbon, nitrogen, oxygen, sulfur. You never, when you add them together, you never get almost never get an integer mass charge. You get some shift, and these fall in very predictable places. So on the precursor level, what you need to do is you need to resolve this thing from this thing, these two hills. But if you get a little bit better, you haven't really increased your resolution until if you get like 50K resolution. Now you can start to resolve things between here, and you can do a better job. But there's a whole plateau there where it doesn't matter. Down the MS level, where you mostly have 1+ ions, now the resolution requirement is even lower. And you'll notice that oftentimes with triple quads, if they're doing SRM, they'll use a Q3 isolation width that's bigger than the Q1. They might use greater than even a Thomson. The other thing going on with proteomics that makes it really well adapted to a low resolution instrument is that most peptides are, you have say like greater than 10 residues. So there's 10 Y ions, 10 D ions that you could possibly form that were 1+ charge. And off of them, lots of them form many of those. And if you try to make an assay, if you try to look at these peptides and say how many good transitions do they have that don't have any interferences, often it's in the range of 4 to 5 or many more transitions that have no interferences. So this having more fragments gives you an extra degree of selectivity, so that if you have a full scan instrument like an ion trap, you can pick the transition that helps you identify the peptide. The other thing is that it's not just a mass spectrometer in isolation. We have an LC, and knowing the retention time is a very powerful filter. So if we look at where our peptides distributed in MZ space and retention time space for a 30-minute HeLa run, if we didn't consider the retention time at all, then if you looked in a 1 Thomson window, you would have like 100 other overlaps with any peptide. But if you know the retention time to within a peak width, now you only have 1 overlap. So this is a huge filter. And now search algorithms are taking that into account, and that this is very powerful. So my thesis here is that ion trap resolution is more than sufficient for proteomics. Okay, now the last hurdle here that people sometimes hurl at us, or last stone that they'll throw, is that ion traps are bad for quantitation. And this could have been true at some point in the past with the 3D ion traps, and with any kind of trapping device, you can see that if you put more ions than you want in there, then they start to interact, their frequencies change and spread out, and so you get mass shifts, you get broadening. And any kind of mass spectrometer also has some finite range for the detector. So as you put more ions in there, eventually it'll plateau. So these are bad things that can happen. But as Joost pointed out in 2002, even on 3D traps, that with regulation, with a feedback loop that we call automatic gain control, or AGC, he said that they could already do a good job in 2002. So this basic AGC process is a very simple kind of deal, where you would take some previous acquisition. It has a characteristic injection time. We're going to look at what that is. You're letting ions in for an amount of time. You measure how much you get, and then you consider those results with respect to how many ions did you actually want to have. So it's pretty simple. So you wanted 10,000 and you got 5,000 with a certain time. So now you know what to set the injection time to. And this is a video from our simulation program again, and it's showing the kind of gate that we have now since the Tribrids that was invented by Mike Senko. And it's a very interesting device. It allows us to go down to in the microsecond range of gate times by having these two things. We can talk more about how it works later if you want. What we're seeing there is that this gate was on at first. Now it opened up. We're letting everybody through, and now this gate, it turned off. And these are the ions that we're going to measure. So we're going to measure how many ions there are. Maybe say it's 6. And then we're going to divide by how long they were let through there. And this is what gives us like our count rate, our intensity axis. In this process, it greatly expands the dynamic range of concentrations that you can look at. You have one sort of limit being your detector, that saying I have an upper limit before a peak is going to saturate, and you have a lower limit that could be either, we can see with our detectors down to a single ion, but you need more than a single ion to get good quantitation. Because of some statistics, you might need 25 to get like 20%, or maybe you need 100 to overcome like ESI spray instability. So you have this lower limit there. And then you have the range over which you can switch your gate. So we can go down to the microsecond range here. And on the upper limit, you'd be you have a limit based on your LC peaks have a certain range, and you need to sample them. So given all these considerations, this AGC process, it expands your dynamic range to a point where the gate and the trap itself is no longer the limiting factor. The limiting factor is the ESI process. And we can see this if we do these concentration curves, these dilution curves at the high end, they'll roll over and you look at those peaks and you have the correct number of ions. They're not saturated. What's happened is that you don't have enough protons to ionize everything. So the next great advance in the ion trap world was the linear ion trap. Our version of linear ion trap, it was invented in the 90s by these two guys, by Saika and Beer. And it solved the problem of the 3D ion trap of how to get the ions in and how to get them out with high efficiency. And this led to, like almost a decade later, the commercialization of the LTQ. And so we can compare these two devices. They look much, much different, but they operate very similarly. And from the paper in 2002, that was this work was led by Jae Schwartz, our ion trap luminary for many decades, and they showed that the mass shifts that you get as a function of number of ions, they were shallower in the linear ion trap than the 3D ion trap by a factor of 20 or so, which was similar to the increase in volume that you got. So now we can fill these ions, these devices up really high, and we have a regulation means. And so the space charge is not a problem. Quantitation can be very good. I was saying that this device had solved the problem of how to get the ions into the device, into it. So before we weren't able to trap all the ions over the whole phase of RFs, but now we can. So if you compared it to before, the duty cycle was increased tremendously, and this made the linear ion trap way more sensitive. And if we look at the publications, they really start to rise after it was launched and basically completely displaces the 3D ion trap. Okay, so we're gonna, just shake you up a little bit. So at this point in like 2010-ish or so, we got really good, or the field got really good at counting peptides, at making the lists, they had false discovery rates kind of being sorted out, and that was all doing well. But as Borcher says, that wasn't enough. Now biologists wanted to know not just what was in the samples, but how much was in the samples. And the gold standard tool, and I think people can argue that it's the gold standard now, was the triple quad doing selected reaction monitoring, or it's what is called sometimes multiple reaction monitoring when they are looking at multiple fragments. So you're isolating a precursor, you're fragmenting, and you're selecting one of these or multiple of them to monitor. So the features of the proteomics experiments, they were not just qualitative, now they're starting to be more quantitative. They were larger scale. Around this time, we saw such papers as like the 1 hour proteome paper from Kuhn's lab. It wasn't so manual anymore. We're doing nanoflow. We're not doing always the fractionation and the gel electrophoresis. And as I say, the searching was much better. And if you look at like comparison with a triple quad and a linear ion trap for the quantitation that you might want to do, if you just look at a single transition, and if you compare this situation for a triple quad versus a 3D ion trap, it's no comparison. That the triple quad is way better, close to 100% versus 2.5. And if you compare it to 2D ion trap, though, it's getting closer. You could say that it's still a factor of 2 better, but in reality, in almost no experiment is it okay to just monitor a single transition. Like you could do that if you had gone through a bunch of other hurdles, but almost always, even in the small molecule world, you want to have another one that gives you some qualitative information. And papers like this one from Sherman said that a unique signature for a peptide is about 5 transitions. Maybe during development, you would have 5 or more, and maybe you would come down to less, but usually like at least 3 is kind of the minimum for proteomics. Minimum. So this cuts down your duty cycle on a triple quad by about a factor of 3, whereas for a linear ion trap, it doesn't do anything. You still have 50%. So you could say that the sensitivity parity for proteomics between these devices was reached in 2003, but nobody really noticed because of the great reputation and the good results that people got with triple quads. And so ion traps continue to be used in this qualitative role, especially in the hybrid instruments that we were making, like the LTQ Orbitrap, where you'd have the Orbitrap to be doing the precursor selection and accurate mass measurement and the quantification if you were doing DDA on the MS1 level. And so that was kind of summarized here by Doman and Aebersold. You'd have that quantification on the MS1 level with the MS, and then you'd have the identification being done with the ion trap, because we talked about before that you don't need a whole lot of resolution, especially on that MS2 level, to do good identification. And people also got interested though in making things more efficient. And there's an interesting sample multiplexing that you guys all probably know about, the tandem mass tags. And Gigi is and has been the real leader here. And from their paper, they have this nice figure where they were showing if they had this 6 different samples, they could label them with the different types of labels. And when they mix them together, the tags have the nice property that all of the different samples for every peptide would have the same master charge. And then they could fragment this and form unique different channels. And so you could do relative quantitation of your samples. But the sort of elephant in the room, as they said, was that it didn't really work because of the fact that when you tried to isolate this one peptide, you actually would get lots of other peptides that had different ratios, and they would fragment to the same channels, and you would do a poor quantitative job. So Gigi and Ting, they showed this where they were trying to measure these 6 different channels. They're trying to measure these ratios. And so they could do it if there was no interference. If they mixed perfectly together and they knew the ratios of the channels ahead of time, then they could measure accurately a 2.5 to 1, a 4 to 1, and a 10 to 1. But as soon as they put in some other sample that had a different ratio, then it didn't work anymore. And this is still the case today. If you try to do MS2 with TMT, it's not really all that good. But they showed that if you did MS3, if you selected an MS2 fragment, then it's just a very powerful filter. And so you could do a good quantification even with interferences. And then Graham came along and he exploited even further the properties of the ion traps, being able to do MS3 by doing multiple isolation of different MS2 fragments. And so he has this figure that we can kind of walk through. Here's where you have the multi-notch isolation. You were able to take more spectra because it was faster now than if you did just a single isolation. The standard one was much slower, and you were accurate. You got the right ratios compared to with MS2, where it was very fast, but you didn't get the right ratios. And now doing the multi-notch, you got the same amount of reporter ions. The intensity was compared to the MS2. So everything was very good about that. And this was an important experiment also with the next instrument that we launched, the Tribrid, where Senko, with his broad mass spectrometry knowledge, but also with his training in computer science, he was really thinking about multiprocessing and the ability to do multiple things in the instrument at once, to always be accumulating ions. And here this is showing that we're always using the collision cell to accumulate ions while the other detectors are going. And this raised our duty cycle up from like 50% up to 90%, here it's 35 for this experiment length. And so now we have not just a single thread, we have a multi-threaded sort of architecture. And this was a huge leap in sensitivity for the ion trap, about 3 times. And if we compare it to triple quad, you could now say it's like 3 times better if you're looking at a single precursor, but no one still noticed because everybody's still using triple quads for quantification. As all these things were happening with the ion trap instruments and other instruments, the sensitivity of the devices as a whole was really raising. And there's this great paper by Thomas Covey describing how with the SciEx instruments, how there was an increase of 1,000,000-fold in sensitivity from the 70s to 2020. So this was happening with all the vendors' instruments. Especially, I remember when I first came to Thermo, they were introducing the ion. It was the stacked ring ion guide at the time, and there were advances opening up the orifices more. And these increases in sensitivity were changing how long you needed for an experiment. So where you might need 200 milliseconds. I remember when I first came to Thermo, it was a big deal that we could run at 10 hertz. And this is about how long the experiment was. And then in 2013 or so, a typical experiment for the ion trap was about 40 hertz. So this actually has an effect on the duty cycle as well, as we'll see. But what we then noticed as we were playing around in the lab in the mid 2010s is that the instruments were so sensitive, our quads are now so good as well, that with 10 milliseconds of injection time, you could do a really good job. You could do good quantitation and you could do good identification, actually. And so what we did is we inspected and optimized every single process that you can think of, all the transfers from here to there, the transfers from here to there, how fast everything switches, electronics were updated. And we were able to go from a world where the scan rate, originally on the Fusion, it was even less, but the scan rate for a proteomics experiment would be, if you're looking at like a 1200 Dalton range would be 40 to 50 hertz. Now we got it up to 70 to 100 hertz. And we can, if we really, really tune things screaming, we can go faster than 200 hertz on these things. So we were looking at the speed that we could get only because the instruments were more sensitive. And this actually has, because there's some overhead that our electronics didn't quite keep up with that and the ion moving speeds. So you actually lose a little bit of duty cycle because we're going faster now these days, 84 versus 90, but that's not a huge deal. And the triples also feel that as they have switching times as well. And so ion traps are still more sensitive, I'll argue, even for a single precursor. So we can look at what happened with the duty cycle. So we had LCQ, we had 2.5% efficient, then on LTQ went up to 50. It goes down a little as instruments get more sensitive, but no one notices because things are growing by 10x. And then on the Tribrids, we had the great leap, and now we're over here. So let's look at now, let's look at some real world examples that try to prove this assertion I'm making about the sensitivity of the devices. We have a matrix matched dilution curve, which is the way to go to compare lots of different peptides into different samples. And we have our instrument, the triple quad, which is a very good one, a very fast one, compared to our new instrument that we launched this year, the Stellar. You may have seen a plot like this and maybe in a place like Skyline showing the concurrency of the assay, how many transitions are active at a certain time. And in this assay for like 700 peptides, 30 minutes, it was above 600, pushing now the dwell times of the triple quad very low, while the accumulation times for the ion trap are above 10 milliseconds. And I have these two different curves here. On Stellar, as you'll see later, we have new ways of doing the targeted experiment that usually we have even narrower scheduled acquisition windows. So usually we would get even more acquisition time. So now we're really hurting the triple quad. We're pushing it into a place where it doesn't want to be, where these dwell times are approaching the instrument times. So it's getting to be very not efficient if you want to look at higher throughput assays. So if you look at a typical kind of dilution curves for the triple, they were going down with concentration, but they had a lot of scatter because there's not enough ions. Your statistics are bad. And the linear ion trap was much better. So, and this curve here is it's actually a line in a log space. Looking at distributions of the LOQs, most of the triple quad ones were not to 5x dilution yet, whereas they were close to 100 or so for the linear ion trap often. And the limits of detection, though, which is where these lines come into the noise, were a little bit closer. They're like 2 or 3 times better for the trap. And it wasn't just us that was able to show this in the late 90s, early 2000 or late 2010s. Other folks like our collaborators in the McCost lab, they were showing that the linear ion trap could do good quantitation, better than the image current detection in an Orbitrap. For this is for a targeted experiment. It was 2 to 3 times better. And we showed this internally. We had this experiment that we did doing parallel reaction monitoring, where we could, with good precision and accuracy, there were more targets that we could quantify than the Orbitrap, either with a certain assay of like 800 targets, or if we doubled the assay. Other folks were showing similar things, and especially at like low loads in a comparison with the Orbitrap, where at like 10 nanograms, the Orbitrap might get a certain number of IDs, but with the ion trap, you could get more. And there was this other nice paper here where they showed a similar thing. This now had FAMES. And then we had Brian Searle had this paper here where they were also doing quantification at these low levels. And although those plots show that the high load ID rate was not so good, some of the new algorithms that like Chimeras that are able to use predicted peptide spectra and retention times, which we talked about as an important filter, we can now get like kind of world-class kind of numbers of IDs in with doing DIA. Although, so yeah, these numbers, like they used to be unheard of, like when we first started doing this kind of work. So it's really neat what these search algorithms can do. I would say though, in our hands, I'm not sure is, we're very interested in the quantification, and we look carefully at the results that you can get on a stellar with like a PRM versus a 4 Thomson window. And they're just not quite as good with the DIA yet with 4 Thomson. We think we probably have to go to narrower windows, but yeah, that's a thing to work on. So with the final little bit of time here, let's look at the state of proteomics today. We have the big winner in methodologies, the data independent acquisition. You can generate easily these huge lists of peptides, although some can argue that quantification isn't always at the forefront. You have data dependent acquisition often being done by TMT diehards, still others that love and others that just want to do label-free stuff. That's still out there. And you have the targeted experiment, which has been relegated to triple quads and smaller scale assays. And the people that are making money in the contract research organizations and these big labs. And those targeted folks that if I look at the sort of the parameters of that targeted experiment that they're doing it with very high throughput, with like very, very fast runs with very good sensitivity, but the coverage is not there. And it's very hard to scale that kind of targeted experiment traditionally. We have, with our new instrument and with the new software that we've put out, we think we've made like a leap in the targeted performance. And Jenny Van Eyck, she agreed with us when she said that they were really happy back in the day on a triple quad making an hour-long assay for about 100 peptides. And now we can make such an assay in 8 minutes and have very low limits of quantification. And we can also go the other way, and we can scale this in an hour. We can look at 5,000 or 8,000, starting to look like kind of global analyses of samples. So we're really pushing on the coverage, and we're trying to make it feasible to make these assays, because you can't do this in the normal way. One of the ways that we're able to increase our efficiency and increase the sort of usability of the assays is by doing something we call adaptive RT. So it's essentially going to be using narrow windows and adapting the retention times of those acquisition windows. And so this is the standard slide we use to do that. We'll have some control data from a previous run or a discovery run from another instrument like an Exploris or an Astral. So we'll take that data, and then we'll take data on Stellar, and we'll compare the data that we just got. We'll take a snapshot, and we'll compare it with the control, and we'll see where we estimate that the current data are relative to the control. So yeah, we're periodically checking them. We're doing this cross correlation to yield a new a shift estimate, and then we are going to update the targeted schedule. This is this kind of neat little animation that we have where we're looking at the cross correlations now, and we're in the light colors. This is showing us too with the cyan lines where we think the retention time shifts are relative to a control, and the white dots are showing the sort of validation with the skyline. And what we're seeing is that the algorithm is able to, with some reference taken like on a day 1, is able to work for more than a month, and it's able to handle this shift in the column that happened at like day 19 or so. So this now is what we're saying is like making us go from this worry. If you're running a targeted assay, you're worried, you're checking it every few days, you're seeing, is everything shifting, how is it going? And 1,000 targets really would be a nightmare previously, and now we can do it. So where do we go from here? We have a few ideas. Maybe you have other ideas, but we would like to keep pushing on the coverage. We'd like to make this more global, and we'd like to keep making it easier. We'd like to, largely with software things that we could do, but there's also the properties, the MS3, that we could make easier. So we are collaborator Mike McCoss, he's in that genome sciences department, and he gets inspired by some of the things happening there. And he's made this comparison of what happened with DNA sequencing, where the first generation was these long reads, global scope very expensive and slow. And then later, the field was really affected by being able to do shorter reads and a targeted thing. You didn't have to look at the whole genome, but you could go very, very fast, and everything was made less expensive. So it doesn't quite fit in the same way, but you can try to make an analogy with the Discovery MS experiments. Now with DAA, they're getting to be just a few tens of minutes, and you can almost get a global snapshot, but they are more expensive these days. So we are inspired to think about next generation targeted instruments that have targeted scope, limited scope, but they're very short time, they're less expensive, and they could be modular. Users could select what the pathways they're interested in, the proteins of interest, and we could say yes, do those, and maybe they could combine them, and that could be very powerful. I was talking about MS3. This has come up in some of our experiments running up to ASMS, like with Van Eyck. They were interested in this CRP peptide, which it has this interference that you can see with MS3. You know, it wasn't lining up the light and the heavy standard, but with MS3 we were getting 3 times better LOQ. So this is a great thing. You can't do it on a triple quad, and if we have tools, software tools to do MS3, but we need to make it easier to like kind of mix and match them within the assay. So the final thing we'll talk about is just the efficiency of the assay, trying to go towards a global scope. If we look at like an efficiency metric of a targeted assay, you'd have the peak width, which is where you need to acquire data, and you have the acquisition window, as we call it, and you'd want to be only really acquiring data when the peak is eluding. So you'd want to shrink this window if you could. Right now, this is one of our like cross correlation plots showing how there's like this spread in where analytes elute, and our alignment that we're doing right now is really global, but I'm sure one could think of ways, we can think of ways that it could be made more individual for every peptide so that we could just target each one without having to put in standards for each one. So that's something that we think about. And if you think about the efficiency, like where could we go? How much better could we do? The traditional targeted assay might have used acquisition windows of 2 to 4 minutes. We're now using half a minute to a minute typically. And so we've gained, depending on how you compare, like 2 to 8 times efficiency. What if we could go another 2 to 8 times? That could be within reach, and that would take us from 8,000 peptides per hour to like 24. That's like a really global kind of scope. Or maybe in a 20-minute method, we could look at 8,000 peptides. That would be something, I think. And there's other things that you can do with ion traps. We saw that with Brooker, they had this big sensitivity increase, like 5x or so, by always accumulating multiple precursors and then having a fast TOF down there. So that's a very interesting idea. That and ion traps can do something similar. We've shown this at ASMS a few times, but we haven't fully developed it yet, where you could isolate multiple precursors. You could then activate them either with the resonance CID one at a time, or you could do beam type CID, and then you could analyze them. So, and that is it here. This is my summary slide, thinking about where things are going to go. Are we going to make more easier to use methods and to use MS3? Yes. Better mass accuracy? Maybe, but maybe a factor of 2 to 3 we can get with some more like temperature stabilization of the mass accuracy. The resolution, a little trickier to do, but maybe there's an idea or two. Maybe not 10x though. The sample multiplexing, we showed we can do this now in a targeted way. I think this is a very interesting idea to go down. And can we go faster? I think so, yes. More sensitive. People are working on it, right there. And maybe we'll all have to find new jobs pretty soon, though, if the next generation, like the single molecule protein sequencing, comes into play, but maybe not, according to Mike. So with that, I'm all finished, and I'm happy to take any questions that people may have.
Awesome. Thanks, Philip. This is I love seeing your talks because they're really educational. They really push the our as an audience's brains in thinking about these types of things. There have been a couple of questions that have come in through the Q&A. So everyone just kind of answer or ask your questions, and I will post them to Philip orally. So the first one that has come in is asking questions about contrasting ion traps and orbit traps from the perspective of PRM sensitivity. I know that you talked about Lillian Heil and Mike Makas's paper on comparing them from the standpoint of PRM. Can you compare them a little bit more in terms of quantifying, a handful of peptides with deeper sensitivity, and maybe contrast them with TOF or modern TOF approaches at this point?
Yeah, I can try to tell you about some of the factors that come into play. So the orbit traps are great, and since they are like parallel reaction monitoring instruments, they actually have some of the same advantages I was talking about versus a triple. So that's one thing that helps them out. But it's difficult, or it's almost like a miracle that they're able to get the ions into that device through that tiny little hole. So that thing has some efficiency that I wouldn't be able to quote the exact right number, but it's not the 80-90% that is to get ions into an ion trap. So that's one thing. And then the image current detection. There's people can argue about exactly what the lower limit is, but it's not one ion. It's some factor greater than that. So those are the two like main things that are helping out the ion trap if you do a sensitivity comparison. So now, yeah, I mean, with DIA, of course, you try to make up for that by doing some multiplexing by having multiple precursors at once. And I maybe haven't personally done the comparison of DIA on an Orbitrap versus PRM on an ion trap to know exactly how it goes, but I think that it's, yeah, I'm not sure about that. So, so we also were talking about TOFs then now. So the modern TOFs, they don't have as much of the problem with the duty cycle. Like if you talk about the Xenotoph, or you talk about the Astral, you have like this collection of all the ions, and then you're almost always can be filling one place while you're analyzing the other. So they can now be approaching the same kind of duty cycles as a linear ion trap. So I think you could say all of the vendors are probably maybe equally good at getting ions from one place to another once they're in the system. So there might be yeah, they might be approaching some kind of parity there.
Yeah, it's an interesting point. Yeah, I think all of the vendors have gotten at least a little bit better at managing ions inside of the instrument now. We have another question, which is for stellar lit data. Do we have a recommendation for free tools to analyze this type of data? I know that the tools that our lab produces are free and viable for that, but I think that there are a bunch of others. Do you have recommendations?
I haven't been able to look at it yet myself, but I know Macosta said that MS Fragger is another one that does pretty well. I think
Mhm.
as a commercial person, I'd have to pay for it, but it's freely available for others. Um, we've been using Sequest for such a long time, but I don't think that it was using, and it was fine, but we saw this huge jump using Chimeras and some of the new tools. And maybe like if you're using like things like retention time and your tool as well. But I think that's important.
Yeah. What do you think about, so you mentioned that there was like a 13% sacrifice in isolation, moving from high resolution to unit resolution. Do you think that there's a world in which we try to treat detection and quant sort of separately? Like, is there any logic in trying to separate out the two types of methods where we would do, say, detection on high resolution instruments and quantify on low resolution instruments, where you've clearly shown that quantification works well there?
So you're saying why have two instruments to do that in the way
Well,
that
why
was
not
argued?
have two ins? Why not sort of treat those types of experiments as being completely separate?
Well, yeah, I guess with the only problem with doing it in the orbit trap and the way it had been in the past is it was an MS1 level thing, and so I don't know that I'm not sure, you might know better than me, but I don't know that the quant is so great in the MS1 level. So although you could detect something, maybe the quantification wouldn't be quite as good. I know that's something that Mike would also harp on. But I'd also heard people talk about just the fact that right now, even people will mostly talk about in the DIA world just these how many things I've identified, but it's maybe more important to say like how many things are actually like measurable somehow, not just like are in my sample, but are measurable. And I know I saw there was a preprint from the Van Eyck lab with Jing Fu, where she was trying to look at that kind of thing in DIA. Like, yeah, yes, I've identified it, but now really carefully which ones actually have good variability, of which ones can be measured. I would like to see some kind of definition of a quantifiable detection.
Yeah, I completely agree. I think that that's a critical point, which is that not every peptide that we can detect is actually really measurable in a linear way. There's a couple of questions in commenting on the role of deep learning tools like Prosit inside of Skyline, for example, for building these types of methods. Do you want to talk about how they have changed the game with respect to analyzing ion trap data?
Yeah, I think they're really critical because if you're, for one thing, I didn't talk about it, but if you're making like a traditional absolute quantitation assay, the first step is not like it's like an ID step, but it's not like the traditional one. You put in your standards, and Prosit can like so very accurately predict what are the distributions of not, you know, not of the master charge, but the intensities, and so you can like almost unambiguously find all of your standards. Like even like for the PQ 500, where you have 800 standards, you can find them all, and it just takes then like a few runs, and you already have an assay made. So that is really neat. And I know that you've probably seen the papers out there where they show that like using such a spectral library, an in silico one, is actually better than doing often an experimental library, because the experimental one, it may have noise, it may have multiple peptides in there. So that's very important. And what I like to think about is how these tools, these deep learning tools, they kind of like help an ion trap maybe relatively that has the lower resolution. It helps us more than the other instruments that already are leaning on this mass accuracy kind of crutch. So it's making the lower, the more like entry level tools, the less expensive tools, it's making them more and more powerful. And so I love a thing like that. And it's yeah, that's really neat.
Yeah, I think that's a very important point. I think that has really opened the door to making linear ion traps be more viable longer term, and being able to tolerate the low resolution is having kind of at hand accurate fragmentation prediction and retention time prediction. Sandy asks, what is your perspective on the future for single molecule measurements with mass spec? I guess in particular with ion traps, and then if you want to comment also on single cell as well, because I think that those are two very different questions.
Okay. Well, again, you know, I started to get into applications, but I only hear people say, like some people say in 5 years, everybody's going to be doing single cell. Like that's the only way to go because of the arguments about you really need to know what is the heterogeneity. So those are things that, you know, you would be able to answer more. I know that people have, I know that some of the requirements for single cell is you have to measure, say, thousands of cells. You have to get any kind of statistical power. And so for this, you're going to need a very fast measurement. You're going to need something that's very sensitive, of course, because now you're at low levels. And so we're trying to do this in a targeted way. And you're also trying, Brian, to do this in a targeted way. And that has lots of things that, lots of advantages. If we can build these modular assays that other people can use, it could be an accessible way to measure thousands of cells.
Yeah, I 100% agree. I know that there's been a lot of work on single molecule analysis with orbit traps. Is that even feasible with ion traps? What do you think?
I know we've had people come in and do demos like the Ryan Kelly lab, so they think it's feasible. You there, I'm not sure if the goal is always just to quantify everything or to be able to just tell the difference between cells. And so the burden on like on the coverage may not be as high. Like you may be able to just measure 100 proteins and do a good job. And so they're being able to do it in a targeted way to sit on the things that are interested in accumulate is going to have an advantage over doing a DA kind of unbiased approach.
Okay, excellent. So I think that there's a question in the chat now about on making measurements with targeted quants using very small number of targets. So if you were to go down to say 10 peptides or less, is there an argument for opting for an ion trap over a triple quad in those circumstances?
Yeah, as you approach the limit of having few things, then now the efficiency of them, they start to look the same. So the sensitivity wise, they can be the same. So then the argument becomes more on like how long did it take you to set up that assay, or what how are you doing your data processing? With a triple, you have to choose which transitions you look at ahead of time. And I know I hear horror stories about people like in as they move towards the clinic of having to choose, you've already done these months of work and now something's happened and you have an interference. You have less ways to deal with that with the triple quad. With ion traps, we have also MS3, which can give us selectivity that rivals the high resolution accurate mass. So in those cases, I say, yeah, we have other things that can make our instrument better.
Yeah, I think that that's a really interesting point. That there's other factors other than just sort of speed. The ease of implementation and then extensibility of implementation is definitely a serious consideration. I think that I guess from that perspective, how do you see ion traps in use in clinical settings, where triple quad's still king in those circumstances? I think that there certainly is obviously an advantage in terms of being able to make the assays be more flexible to challenges or to problems with background, but at the same time, a lot of those need to be really nailed down up front anyway.
That's true. They take a lot longer to make those assays. So it, I'm not sure. What we're hoping is that we have this instrument that it's so much easier to make the assays and the type of results that you can get for small numbers of ions is very similar. So why not just keep it there? The technology is so similar to a triple quad in many ways that hopefully those clinical researchers will say, yeah, same, but it cost the same or less, and it was it costs less time to do this thing. So that should surely be some kind of consideration.
Yeah, very cool. So Devin Drew in the chat asks about says, adaptive RT is awesome. What do you think about are there limits to what it can do with regards to LC variation? And are there things that we need to kind of consider in terms of how much correction is applicable and over how many days of acquisition time?
Yeah. Yeah, so some of those, you saw we went to a month and we went to a column change and like lots of trap changes, but we haven't seen like, can it last a year? Can it do, I'm not really sure yet. We're starting those experiments now, so we hope to have more information about that. I will say that as we did like lots of different gradient lengths, at first we started out with an hour long, and that's has one set of challenges. Then we went to these 8-minute gradients, and there was another set of challenges. Like we weren't really ready for like how fast things change. So right now, sometimes we can have problems like at the very beginning of an assay. There's not a whole lot of information, and but usually there's not a whole lot of targets either. So our approach is we want to just increase the size of the acquisition window at the beginning, and but that at the beginning or at the end, that's a place where you could have an issue, and because sort of the strength and the problem is that we're using just whatever is eluting. We don't know exactly what it is, but so if there's nothing really eluting, we're not going to have a lot of information about when it comes out. But we have seen it be able to take, LCs can do really funky things, and the way that people use them these days, they often have like a really high flow rate at the beginning and the end to get that equilibration done, and this causes these funny kinks and things happening um, in the shifts of molecules that you can't really solve with just like a linear thing correction.
Great. So we got one more minute for the hour. I just had one last question. What do you see as the role of triple quads moving forward?
Well, yeah, you got to say like they're still great tools, and you have to consider not only what it can do, but how much it costs, and like the price per performance thing. It's there. Like it's like we haven't gotten rid of Honda Civics because there's still some of that was going to want to use them, and yeah, so there's a place for everybody, I think.
Hmm. Okay, well, we're at the hour now. I want to thank you so much, Philip, for giving us this excellent talk. I think everyone has really appreciated it. So I'm going to close this session of the HUPO Education and Training Committee webinar series. And I want to welcome you guys to come see the next one. Fantastic talk. Thanks again, Philip.
Thank you. Yeah, bye-bye.